As per PDA Technical Report number 34, isolators are defined as a decontaminated unit meets ISO Class 5 condition, provide continuous uncompromised isolation of its interior from surrounding environment. Isolators represent the highest level of containment, and are widely used in sterile manufacturing processes where product contamination must be avoided. Isolators are majorly divided into closed and open isolators. Closed isolators do not exchange unfiltered air or contaminants, or any material flow with adjacent environments. Examples are sterility testing isolators and cell gene therapy isolators. Open isolators are designed to allow the continuous or semi-continuous ingress and/or egress of materials during operation. Examples are isolators used to fill vials, PFS, and cartridges. Let's talk about open isolators system in this video. The major component of isolators are isolator chamber integrated with filling machine, upstream and downstream machines, air handling and filtration system, decontamination unit, glove ports, rapid transfer system and environmental monitoring system. Air handling and filtration system. Isolators rely on a sophisticated air handling system that includes HEPA filters to ensure that air entering and exiting the isolator is free from contaminants. For aseptic processes, the isolator environment is maintained under positive pressure. A separate venting system may be provided to allow for safe and rapid removal of the decontamination agent. A decontamination unit required to convert hydrogen peroxide into vapor formed through a heat plate. Liquid vaporized hydrogen peroxide is widely used to decontaminate the inside of pharmaceutical isolators. Liquid hydrogen peroxide is evaporated outside of the isolator in a decontamination unit, which carries the vapor with air into the isolator, where it is recirculated. Nascent oxygen generated during this process is a powerful oxidizer that kills bacteria by damaging their biological functions. Glove ports. One of the defining features of isolators. Glove ports allow operators to manipulate the equipment and materials inside the isolator without breaking containment. The gloves are typically made of materials resistant to chemicals and pathogens such as neoprene, EPDM and chlorosulfonated polyethylene. These gloves required to be tested for integrity before and after use in situ condition. Rapid transfer ports are used to introduce or remove materials and equipment into and out of the isolator without compromising the sterile or contained environment. These ports allow the movement of components while maintaining containment. Isolators are equipped with online continuous monitoring systems for viable and non-viable particulate materials to ensure the state of environmental control. Let's talk about different steps of VHP cycle. Glove integrity testing of all gloves is performed before start of the isolator loading. Then isolator chamber is loaded as per qualified loading pattern with the required articles such as microbial monitoring media plates, sterilized articles such as rubber stoppers, bowls, scissors, tweezers, wipes, etc. Isolators doors are closed after loading of articles, and the isolator chamber is tested for the leak rate via pressure decay. Even open isolators must be capable of being rendered sufficiently leak-tight during the decontamination process so that occupational safety and health requirements can be met. After successful isolator leak test, all the gloves are either inflated using air or arranged in a manner that entire surface of gloves can be exposed in VHP contact. Finally, liquid H2O2 container is loaded inside the decontamination unit. VHP cycle started on isolator HMI once all prerequisites met. Preparation phase is the first phase of a VHP cycle where isolator achieve the predefined temperature and relative humidity value as per recipe set points, and reheating of peroxide duct work takes place. Conditioning phase is the second phase where H2O2 is injected with a high rate from decontamination unit to achieve a high gas concentration to achieve the predefined limits. Decontamination phase is the third phase where H2O2 is injected at a lower rate to replace inactivated peroxide and keep level. H2O2 concentration is maintained constant for a predefined time.
The filling machine are designed to run in creep slow mode to expose all inaccessible parts under VHP exposure. H2O2 vapor recirculation starts at this phase through HEPA filter plenum.
Aeration phase is the last phase where H2O2 is flushed out from all isolator surfaces including filters by air handling unit and once through mode. The time depends on both air exchange rate and H2O2 desorption from the decontaminated material. The final H2O2 concentration is accepted below 1 ppm as per